All right, ladies and gentlemen, welcome back. So we are going to start getting into the actual processes of transcription and translation. But before we do, let's just kind of do a little bit more review of just big picture, right? So when we talk about transcription, right, transcribing or copying, we're creating that mRNA molecule based on the DNA code, right? We're copying that section of DNA, that set of instructions for that particular gene. And the RNA molecule that we actually create is messenger RNA. So it is your chemical message. It's the product of that transcription. Now we'll talk about things like transfer RNA and ribosomal RNA, which also get involved here in this process. When we talk about the flow of information, we talk about really DNA to messenger RNA, and then we get to the proteins. And those proteins are being created via translation. So now we're going to use that message in that messenger RNA. That messenger RNA gets made in the nucleus, leaves that nucleus, goes to the ribosomes out in the cytoplasm, and then actually gets translated. We're translating from one chemical language of nucleotides into another chemical language of amino acids to build that polypeptide, to build that protein. And all of that translation is happening at those ribosomes. Now, when we look at transcription and translation in prokaryotic cells versus eukaryotic cells, the process does look a little bit different, and it really comes down to the fact that those prokaryotes don't have a nucleus. So that messenger RNA is getting produced via transcription, and as soon as it's done, even before it's finished, we can already start attaching those ribosomes and doing translation. So this can really be a continuous process, which means that it's relatively quick. But it also then does hold to one gene, one protein. There's nothing that's happening in between that. When we look at this process in eukaryotic cells, on the other hand, that nuclear envelope does complicate things, but on the flip side, it does in some ways make the use of information a little bit more efficient. So transcription is going to have to happen in its entirety inside the nucleus of that cell before we can export it out to the ribosomes and the cytoplasm to do translation. But what does happen between transcription and translation in eukaryotic cells is that oftentimes we will go through this RNA processing step. And that RNA processing in certain situations does also allow for what we'll talk about as alternative splicing, so that we can sort of mix up some components of that gene and not necessarily use all of it or not necessarily use it all in the same order to make similarly functioning proteins, but they're not identical. Regardless of whether we're talking prokaryote or eukaryote, though, we do go through this idea of this central dogma. So we talk about some major theories, major ideas in biology. We've talked evolution, we've talked the cell theory. Central dogma is another big one. And it's, again, just that flow of information that we keep kind of referencing back to, that your DNA holds all of those initial instructions, but then we'll just take a copy of the piece that we need for that particular situation into that RNA, and then that RNA is what goes to the ribosomes to actually get translated into proteins. Okay, so let's talk through transcription here. This is the first stage of actual gene expression. So we've talked through DNA replication. How do I make copies of the instructions? But now let's talk about how we use them. So that RNA synthesis, that messenger RNA, needs to be catalyzed by an enzyme, just like our need to use the enzymes in DNA replication to make more DNA. Now, good news is that the number of enzymes you need to remember here are a lot less than the number of enzymes we talked about when we talked about DNA replication. So the only major enzyme that we're really going to focus on here with transcription is RNA polymerase. Okay? RNA polymerase is going to do a couple of jobs. So it's going to help pry apart the DNA strands, right? So basically snip those hydrogen bonds that exist between the nucleotides in DNA so that we can get access to the code. And then it will read the template strand of DNA to then hook together RNA nucleotides in complementary bases. Now, something to remember is that when we talked about base pairing in DNA, right, it was A with T, C with G. There is no thymine in RNA. Instead, it's a uracil. So base pairing rules become a little different here, so that it is A with U and C with G instead. Now, here's another component we have to understand is the actual gene structure. You've got 46 chromosomes worth of information packed into your nuclei. Each of those individual chromosomes may have 10, 20, 30, who knows, number of genes on that individual chromosome. I'm not going to be accessing all 10 or all 20 or all 30 genes on that chromosome at any given point in time. So how do your cells know where a gene starts and where it stops? Well, sort of think of it like a textbook, right? How do you know where a chapter starts and ends? Title pages, review questions at the end. There are different section numbers, headings in the middle, right? And the rest of the actual information you're asked to obtain is in there. So that's kind of how this works. We could even go simpler and just talk about a sentence. How do you know where a sentence starts and stops? You've got a capital letter at the beginning, a punctuation at the end, and the actual content of the sentence is in the middle. It's kind of how a gene works. You have a promoter sequence, all right, which is often common to that particular species or particular set of genes in that species, a sequence that your cells recognize to know that this is the start. You have the actual transcription unit or an RNA coding sequence in the middle. That's the actual DNA that's going to get transcribed and then ultimately contains the information that will get translated into proteins. And then you have a terminator sequence, again, often common among different genes in the same species or sometimes even across species, to know where the signal ends, where the sequence is done. So kind of think of that promoter as the capital letter, right? That's how you have been taught. That's how we have decided to illustrate that this is the beginning of a new idea. Terminator would be your punctuation, the period, the question mark, the exclamation point at the end of the sentence. That's how you know the thought's done. And then the RNA coding sequence in the middle is the actual meat of the sentence. What is it actually telling you? Something else I'm going to point out here while we're talking about this, because I'm going to use this terminology and it's definitely going to come up as we continue talking this unit, is the idea of being upstream versus downstream. All right, so upstream would be towards the promoter end of the region, terminator would be downstream of that, and then your RNA coding is in the middle. And also pay attention to where the 5' and 3' are as well. So let's talk through the stages of transcription then. So now that we have the basic understanding of what transcription and translation are and the basic structure of a gene, let's talk about making copies of it. So we can break transcription down, the actual process itself down, into basically 3 major steps: the initiation, the elongation, and then the termination of it. So initiation is just going to be that RNA polymerase molecule being signaled to go attach to a particular promoter sequence for a gene. It's going to attach to that promoter and start breaking up those hydrogen bonds and allowing access to the gene itself, allowing access to the DNA. Elongation, then, we're going to move that RNA polymerase along the gene, along that coding sequence, and start using the template strand of DNA to now build the messenger RNA. So complementary base pairing the DNA nucleotide with its corresponding RNA nucleotide. And that elongation will keep happening until that RNA polymerase hits the terminator sequence, Once it hits the terminator sequence, that signals that RNA polymerase to basically disintegrate, fall apart into component pieces. DNA winds itself back up. Messenger RNA also gets released at the same time. So let's dig into this a little deeper. So if I look at initiation, right, promoters are the signals to tell RNA polymerase where the start of the gene is, right? There are transcription factors, other proteins that your cells create that help to mediate the binding of that RNA polymerase. So RNA polymerase doesn't really do a good job of sticking to these promoter regions and actually starting to open up that DNA molecule for transcription to occur without these transcription factors. So they need to be bound to that promoter region first, and then the RNA polymerase basically nestles itself on top of them and can start the process. So in order for this all to work, we need to build this complete transcription initiation complex. So the RNA polymerase plus those transcription factors will then allow for the next step of transcription to occur. I mentioned to you before, there is often a common sequence in that promoter region. And something we see pretty commonly across all eukaryotes is this idea of a Tata-box. And so it's the T, A, T, and a stretch of A's, and then the reverse would be on the 3' end, going the opposite direction. And so we often find that at the beginning of the promoter region, there may be some other components here as well, but that is a pretty common sequence that we see in eukaryotic promoter regions. Once that initiation complex is all built, then we can start moving down the DNA and actually building the messenger RNA molecule. So that's the elongation piece. So that RNA polymerase will open up the DNA, untwist the double helix, and start adding bases, about 10 to 20 bases using the complementary sequencing at a time. When we look at eukaryotes, we can actually do this, generally speaking, with pretty good accuracy at about 40 nucleotides per second. So it's pretty quick. And cool thing is that that gene can be transcribed simultaneously by several RNA polymerases all at once. So if I really need to, I'll use this term upregulate, meaning I need to make lots of it because I need a lot of the protein, I can actually make multiple messenger RNA molecules almost simultaneously by just stacking RNA polymerase molecules one behind the other and making a bunch of messenger RNA at once. That can then all go out to the ribosomes in the cytoplasm and start making that protein all at once as well. Termination then, mechanism looks a little bit different when we get to that terminating sequence when we're talking about things like bacteria versus eukaryotes. When we talk prokaryotes, bacteria, pretty much as soon as we hit that transcription terminator region, everything falls off, we're done. Eukaryotes, for whatever reason, that polymerase seems to continue on through that terminator region. And then a handful of bases, sometimes 100 or so, past that terminator, which then does need to get edited off once we go to producing the sort of what we'll talk about as mature messenger RNA as opposed to pre-mRNA. So when we talk about the molecule that comes off of transcription initially, it's what we call pre-mRNA. We're going to do some editing to it, do some processing to it. And one of the things that we'll do is we'll take off that excess that got transcribed after the terminator region, even when it wasn't necessary. Eventually, the polymerase just kind of falls off. And the thought here is that because we're not doing this continuously, we're not continuously making messenger RNA that is already getting translated, that there's some benefit to going past the end of that RNA coding region just to ensure that we have all of the information.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is